Hi, this is an example of our blood typing system that I want to show you. If you look at this chart here, you see that we have red blood cells. And red blood cells have on the surface antigens, which are proteins. And the particular ones we're after today are the A and B antigen. And the A looks like this, a little purple circles here, and it's attached to the surface of the red blood cell. B looks like this, these little shapes, which are kind of greenish. That's the B antigen. When the two of them are together, you have both of them on here like this, the AB. And if the red blood cell has neither on them, it would be called O. Now the trick here is, if you look down here, you see that the A antigen has B antibodies, and they are shaped like this because they fit the B. The B has the A antibodies because they fit the A antigen. See how they fit? So they fit the opposite of each other. That's why you cannot give A blood to a B person, because this will attach to that one and clump it. And the same thing about B with the B antibodies. They will attach and clump it, and it will kill the baby. In this case, you have both antigens, so you cannot have any antibodies. So this person has no antibodies. O, on the other hand, has neither antigen, and it has both antibodies, both A and B antibodies. So that's the story of why we type blood. All right, now we're going to do the blood type. We use this card right here, which is called the blood type card. Okay. It has 3 blood wells, and then the antisera here, here, and here. So I'm going to select one of the blood groups here, and this will be sample number 1. I don't know what it is yet, but we'll find out. Put a drop here, a drop here, a drop here. And we take the anti-A serum, which was the blue one, and we put it right beside of that drop, 1 drop. And then we take the anti-B serum, and drop it right beside of it. Then we take the anti-D serum, which is really an RH factor. Drop it here. We take toothpicks. We get 3 different toothpicks. And we stir them very carefully. Don't flick it. Stir it very carefully. Like this. And you see it, if it starts to coagulate, like you see it doing right here, that means it's got the A antigen. So that would be that one. And then let's try the B. If it clumps, it means you have the B antigen. It does not clump. Let's try the RH factor, the D antigen. It doesn't clump. So what this card represents, after about 30 seconds, this card represents a type A blood because it clumped only on the anti-A serum. If it had clumped on B, it would be anti-B serum, it would be B blood. If it had clumped on both, it'd be AB. If it clumped on neither of them, it would have been O. This would be the RH factor. So this is a negative, did not clump, so this would be RH negative. Okay, we're going to examine here the constituents of the blood. First of all, you see that we take a little sample of blood, put it in a test tube, centrifuge it, so that the heavier elements or parts of the blood will settle to the bottom. The lighter will stay on top. And notice you get in the central tube there, you get a separation of the heavy and the lighter material. The lighter stuff will be the plasma, and the heavier stuff will be the formed elements. Now the formed elements are red blood cells, white blood cells, and platelets. In the plasma, you will have, most of it will be water, and then there'll be lots of salts, electrolytic salts, as you see there, and you'll have a lot of plasma proteins, albumin being a very important one. They help to maintain osmotic balance in the body. There are substances also in the plasma that are transported, such as the stuff that we eat, glucose, fatty acids, amino acids, vitamins, and waste products such as urea and uric acid, and some of the respiratory gases such as oxygen and carbon dioxide, and of course hormones. In the formed elements, notice the numbers that you see there. In red blood cells, the numbers should be between 4 to 6 million. And in the white blood cells, they should be between 4,800 and around 10,000. And in the platelets, it should be under 300,000. Now these are the cells that you will see in the blood. This is, of course, an erythrocyte, which means red blood cell. Now, in the white blood cells, you will have, the first one you'll see, it will be the neutrophil. Notice it has 3 lobes. This is neutrophil. Then you'll have the eosinophil, which will have little red dots in there, and those dots are from the word eosin, which means red in biology, so you can tell that one quite readily. Basophil looks like bees' wings. I remember that from bees, starting to bee. So basophil would be your next white blood cell. Now those 3 that we just did are granulocytes. They have grains in them. The agranulocytes starts with a very small cell called the lymphocyte, and then a very large cell called the monocyte. Now those 2 are agranulocytes. This would be an example of a platelet or platelets. When looking at a blood smear on a slide, it will look much like this, where you see the pink cells there are the red blood cells, and the purple ones there, you see purple nuclei, will be the white blood cells. There you notice some of the more popular ones, eosinophil, and lymphocyte, and the monocyte, and the neutrophils. And then you see the tiny little granules, purple granules. Those are platelets. All right, this is kind of a summary of the functions of the white blood cells. Notice that the top 3 of the granulocytes are the neutrophil, the eosinophil, and basophil. And notice that the numbers that they are supposed to have. And then to the extreme right, you notice what the function of these cells are. So, for instance, in the eosinophil, if you would have an excess of those, then you can look over and see that it has to do with killing parasitic worms, and also having to do with allergic reactions and things like asthma too. So that would be, these are very diagnostic things for doctors to check the amount of cells you have. At the very top, you have the erythrocytes, the red blood cells, and notice you have 4 to 6 million of those per cubic millimeter. Now, a cubic millimeter is about the size of your pencil lead at the very tip of your pencil, very small. So you have that many cells. At the very bottom, you see the platelets, and you see the numbers there, and that has a lot to do with blood clotting.